Yeah, I know. No, I don't want an AI. Everything's got an AI companion now. Do you want an AI companion? No, I don't need that. I saw that your sister submitted her first college application. Yeah, excited. JMU's a good school. It's a very good school. I've been there. Mm, I've been there. Actually, one of our very good, very closest friends from like high school, he he went to JMU and met his wife there, and then they got married in Harrisonburg. Like they actually got married like 2 days or a day or whatever before my college graduation. And I had to go to the wedding, and so did Jason. And so I ended up having to miss my graduation rehearsal because I needed to be in Harrisonburg, so I had to get special permission. And then I drove back as soon as the reception was over. I drove back from Harrisonburg to Roanoke so that I could get up the next morning pretty much and graduate. It was pretty wild. It was pretty wild, but I didn't think she was gonna let me go, but my advisor was always good, and she's like, "Yes, you can go. You can go. It's not hard. Follow everybody else." Probably helped that my name was at the end of the alphabet, so I just follow the people in front of me. Had I been the first person in my class, probably would have been. You gotta lead the line, right? Yes. So you girls have worked on your dragons on the side just for fun. Did you breed your dragons together? No? Okay, so feel free to ask one of your lab mates if they would want to breed their dragon with yours. If you don't want to breed them to each other, that's totally fine. Because just kind of something we're doing like on the side as we go. Like you're not going to turn it in. It's just for fun. It's to help you kind of understand the way alleles work and Mendelian inheritance with dominant recessive genes and things like that. So if you want to pass it on to somebody else on your team and say, hey, would you breed your dragon with me? I bet somebody would. You just gotta find somebody who will. Let's work it out and it'll be good. All right. Where's Evan? I don't know where Evan is. We had our church trunk or treat on Saturday and it was fun. We had a really good time. We always have a chili tasting competition. Do you like chili? Do you like chili? Do you like chili? Don't know. Not sure if you like chili. We have a chili tasting competition every year at church. And the grand prize is you get an apron that says that you're the chili cook-off champion in the year. And people want those aprons so bad. They get very competitive about their chilies. I've entered one every year and I've never won. So it's kind of fun though to go and taste them all. They're all really different, even though they're all chili, but they all taste very different. All right, well, I don't see Evan yet, but I imagine he's going to walk in the door any second. But we have a lot to cover, so we'll start. So, like I was saying, it isn't actually Tinker Day, but Tinker Day has got to be sometime between now and Friday. And there's rain in Roanoke today, tomorrow, and Thursday. So I'm thinking it's Friday. They're going to hike the mountain in the mud, I suppose. They probably should have done it yesterday. I was really hoping for it yesterday. But we'll have our donuts today. At one point during y'all's class, I'm going to step over and pop those donuts in the oven so that they can warm up a little bit because they're much better warm, right? Krispy Kreme is better warm. Got your binders? You got your binder. Are you ready? All right, let's go ahead and start. So we're going to start by talking about RNA. So like RNA, like DNA, is a nucleic acid, and it consists of a long chain of nucleotides. So how is it different? There are three important differences between RNA and DNA. So what's the sugar in RNA? Do you know? If it's deoxyribose in DNA, do you know what it is in RNA? Do you know? Emily? Do you know? Yes, you got it. Ribose. It's just ribose. So it's not deoxyribose. It's just ribose. So it's ribonucleic acid and not deoxyribonucleic acid. Very good. All right. Next thing would be that RNA is generally what kind of strand, Emerson? It is a single, not a double strand. Very good. So DNA has double strand and RNA is a single strand, and those little nucleotides are just hanging out there on their own. And RNA contains what in place of thymine? Emerson, uracil. Okay, so there are no T's in RNA, right? There are U's in RNA. And so what would you go with? What's the complementary, Everly? Hey, good, adenine. All right, so you can think of an RNA molecule as like a disposable copy of a segment of DNA, a working copy of a single gene. RNA molecules are involved in what kind of synthesis? Synthesis means they're making something. So what is RNA involved in making? Do we know? Do either of you know? It's going to be, proteins. So this is how we make our proteins. Now, everything that, a lot of things, almost everything that makes you has to do with proteins. So this is why DNA is important, because DNA is going to code for RNA and RNA is going to code for the protein. So RNA controls the assembly of what into proteins? What make up proteins? What are the building blocks of proteins? Little tiny units. Don't know? Okay, these are amino acids. So you see them right here? Here's a string of amino acids. So when you string amino acids together, you get this big, long polypeptide chain. And when it'll fold itself up, and that's what makes your protein. It folds itself up. And it has a certain shape when it's done, and the shape is important. So 3 main types of RNA. We have one called messenger RNA or mRNA. And what does that do? Do you know what it does? You know what messenger RNA does? What's he doing right there? What does he look like he's doing? Yeah, he's delivering a package to somebody. So yeah, so messenger RNA carries information from DNA to other parts of the cell. So it carries information. It delivers a package. You can think of it like that. It's carrying information. Okay, making sure that wasn't Evan. All right, now, then you have ribosomal RNA or rRNA. And you notice that it's a lowercase letter in front, right? There's a lowercase m for messenger RNA, a lowercase r for ribosomal RNA. Do you know what this one does? This one makes up the subunits that make up a ribosome. So this is how a ribosome is built, is with rRNA. And then lastly, we have transfer RNA or tRNA. And do you know what that does? What's it look like he's doing? What's he doing? He's carrying a suitcase, so he's probably traveling, right? So transfer RNA is going to take the protein that's been built in the RNA and transfer each amino acid to the ribosome. So he's going to, when a protein is built, a transfer RNA molecule is going to transfer each amino acid to the ribosome. So you have your 3 main types of RNA here. All right, now, RNA synthesis. How does a cell make RNA? Remember, to synthesize means to create. So how does a cell create RNA? So in transcription, segments of DNA serve as templates to produce complementary RNA molecules. The base sequences of transcribed RNA complement the base sequences on a DNA template. What does complement mean in this case? It's not compliment. That's hi, you look beautiful. I'm giving you a compliment. This is a complement. Do we know what that is? So this is what is meant by C going with G, A going with U. That's what we mean by complement. Whoever it partners up with. Are you girls chilly? No, you're not. 68. It's not that chill. I was gonna say, do I need to turn the fireplace on? But it's not. It's just generally cold and yucky, right? Did you see Jamaica though? Did you see what's happening in Jamaica? Big Category 5 hurricane. One of the strongest ever on record is beating the heck out of them right now. It is not a good situation. All right, so let's look at amoeba sisters. RNA versus DNA. They'll show you the differences that we just went over and summarize them for you. Of course, we're going to get an ad. Yes. Ranger. There we go.
It feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emoji. DNA stores genetic
It's an
information and
emoji.
codes for your trait. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have three parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and in focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree. That helps you remember that A goes with T, and car in the garage. So C goes with G to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different ones? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, more abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles. But we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we?
All right, let's
Pause
see.
after the question so you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have?
Okay, what do you think? 8 nucleotides is how many bases? How many bases? There's a whole nucleotide. How many bases there? 1, she says. Very good.
And also, how many base pairs?
And how many pairs would you have if you have 1 nucleotide? How many pairs of bases would you have? If you have 1 base, then how many pairs? Not a whole pair, right? Half a pair, right?
The answer. Each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases.
Right?
DNA
1
bases
each.
pair like this. And that's 4 DNA base pairs.
4 pairs.
Question 2.
Get it?
If one strand of DNA has these bases shown here, ATTGAC, can you complete what the complementary DNA bases would be for the other DNA strand?
Okay, so if it's a DNA strand, away. A is going to go with T. T is going to go with A. What's going to go here? A. What's going to go here? C, T, G. Got it.
The answer. So remember those.
You did it. Perfect.
For DNA. And the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases?
Okay, what's our RNA going to be? What's going to go here? U, then A, C, U, G, right? You got it. You girls have it.
The answer. So remember,
we did that last year too,
RNA.
so you're familiar
no
with it.
No thymine here, it's uracil instead.
Perfect.
Apples under helps you remember A with U. R in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well,
all right, there we go. All right, so let's look at this. Look familiar? Yeah? All right. So it says you're going to write DNA, RNA, or both. Why don't y'all tell me together? What do you have for number 1? What do you have, Everly? Both. All right. And what do you have for number 2? RNA. What do we have for 3? And 4? There you go. 5? Both. 6? Both. 7? RNA. 8? DNA. 9? DNA. 10? DNA. 11? Both. Good. And 12? RNA. You guys got it. So smart. Excellent. All right, so ribosomes and protein synthesis, the genetic code. What is the genetic code? How is it read? The genetic code is read, as you guys know this because we did this last year at the end of the year in chemistry. How many letters at a time? 3. And what do we call those? 3 letters together. Do you remember? Called a codon. Remember? It's kind of a fun word. So those 3 bases correspond to a single what? What are the 3 bases equal when they're together? What do they code for? A single, remember, an amino acid. So any combination of 3 letters of RNA from their start codon on in chunks of 3, each one codes for a specific amino acid. So proteins are made by joining together amino acids in long chains. What do we call the long chain of amino acids that builds a protein? Do you remember the name for it? It starts with poly because it means many. So it's a polypeptide or a polypeptide chain. So for example, this shows you right here, each of these colors denotes a different amino acid. So there's serine, histidine, and as you go all the way down, this one right here, methionine, you'll find that one's important. That's your start codon, I believe. I think the start or stop, it's one or the other. But it shows you how all those different amino acids will pop together and make a big long chain. So these bases form a language or genetic code with just 4 letters, A, C, G, and U. All right, so look here, because this is what you're going to do in your lab today. You have G, U, A, C in the middle, right? And then each, this is like a pie wedge almost. So if you go out from G, and let's see if you can figure this out. If I have a codon that reads G, U, C, what amino acid am I going to get? Those colored ones are on the outside of the amino acids. It's GUC. Can you tell? So here's how you do it. You find G in the middle. See it? What's the next letter I'm looking for? U. Here's U. What's the next letter I'm looking for? C. Here's C. What amino acid do I get? I think that's valine. Uh-huh. All right. How about if I go C, A, U? What am I going to get? C, A, and then U gives us, that's going to be histidine. And then A of CAA, for example, gives us this one. I think it's glycine. Anyway, these are all abbreviations for the amino acids. So each three-letter word, what do we call those three-letter words again? And there's three letters together. Yeah, codon. Very good. You get these codons. Methionine, there's the one I told you before matters. The methionine codon is start. Do you know what it means to start coding? Anybody want to guess what the methionine is? What three letters? So it's AUG. Do you guys remember when we did that lab last year where we ran around with the cards? AUG meant start. Okay, that's methionine. That means start. So there's no start code in here, right? It's just moving along. So it'll start whenever it sees an AUG and the codon for protein synthesis. And following the start codon, it's going to be read 3 bases at a time until it reaches one of 3 different stop. We have 3 that means stop. UAG right here is one that means stop. Okay, so as it goes along, it's going to code and code and code. So you see alanine, threonine, glutamate, leucine, arginine, serine, stop, right? So what role does the ribosome play in assembling the proteins? Here's a great little adorable graphic of a ribosome and cute little transfer RNA molecules bringing in amino acids. Aren't they cute? Ribosomes use the sequence of codons in mRNA to assemble amino acids. Now, here's something that is very important that you need to understand. We cannot make every amino acid that we need to live. We can synthesize some amino acids, but not all of them. So where do you suppose we get the amino acids that we need to live that we can't make ourselves? You know where we get them? We get them from certain proteins that we have to eat. This is why eating protein is so important, and eating the right kinds of proteins is so important, because if you don't have the right kinds of amino acids, your body can't do this kind of coding. So we eat proteins and it breaks down to amino acids, some of which we cannot make ourselves, and then they are accessible to the transfer RNA. So it's telling it, hey, the ribosome tells the transfer RNA, hey, I need this next amino acid. And then the transfer RNA has to go find that amino acid and bring it down and lock it into the chain. So ribosomes use the sequence of codons in mRNA to assemble amino acids into what? And what are we making? What's this big long chain called? We're putting them into a chain? Polypeptide chain. We're making a polypeptide chain. The decoding of an mRNA message into a protein process is called what? So the process of taking a DNA molecule and creating a complementary RNA molecule for the purpose of building a protein is called transcription. You are transcribing, you are writing it differently. Same information, but you're writing the code a little differently, right? Because you're making RNA from DNA. But the process of making proteins from RNA is called translation, because now it's like you're creating a whole new language. You're producing the protein. It's translation. All right, so here it is. Good old amoeba sisters again will show us how they do it. Oh, yes.
I'm following a teaching degree online.
Yes, yes.
WGU, their respected school of education.
Got it.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on the pigment that is inside the eyes. But in order to have that pigment, you have genes. which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA, well, a few exceptions, and that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like alphabet mnemonics. The transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon. which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA. So the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating.
So there you go. You got it. All right, so let's look here. So this says, for the following discussed RNA types, complete the missing information in the boxes below. Some boxes have been filled for you. So what do you have for 13? What type are we talking about here? We have mRNA in this first column. What's in the middle column? rRNA. And what's in the third column? Number 14. tRNA. All right, so under mRNA, what does mRNA stand for? Messenger RNA. Very good. And what does rRNA stand for? Ribosomal. Good. And then T is for transfer, right? So sketch to help you remember. So you can put whatever you, what did you put in here for 17? An envelope's a great one. Yep. Okay, there you go. And let's see, they made a little ribosome here. And what did you put for 18? What'd you put for over here? Did you draw something? What'd you draw? Anything works, whatever's going to help you remember. What'd you draw? Don't know? Didn't draw? So how about 19? What's the function of mRNA? It carries the message from DNA out of the nucleus. And what's the function of ribosomal RNA? To build the ribosome. It's a major component of the ribosome itself. And then tRNA is going to transfer your amino acids to make your protein. All right, so let's look at this one. So we have a process of transcription and translation, and we need to decide where it takes place, what happens, what's the end result, whether or not DNA was directly involved, and the types of RNA involved. So for transcription, Where does that take place? Yeah, why is it in the nucleus? Why do we know transcription is in the nucleus? Because it involves DNA, and DNA, in eukaryotes at least, DNA is always in the nucleus. So it's going to be in the nucleus. All right, so number 22, what's going on in transcription? Yes, DNA polymerase builds mRNA from DNA. So this is actually the molecule that does the building, but yes, you build mRNA from DNA. Very good. What's the end result? You get an mRNA strand that's complementary to the DNA you started with, right? And then is DNA directly involved here? Yeah, because we're using it. All right, so then in translation, where does translation happen in eukaryotic cells? In the cytoplasm. Very good, because we don't need to be in the nucleus anymore. We need to be in a ribosome. So it's in a ribosome in the cytoplasm. And then what happens in this process? What are we doing? We're making what? Proteins, right? We are bringing amino acids together to match the codons on the mRNA, and then we are creating ourselves a chain of amino acids that will fold into a protein. So our end result is that we have built a protein. Very good. And no, DNA is not directly involved. And what types of mRNA are involved here? All, yeah, tRNA, rRNA, and mRNA. mRNA because that's the strand we're working with. That's our template, right? rRNA because that's basically what composes the ribosome, and then tRNA because that's what's bringing the amino acids into the codons. Very good. So let's talk about mutations. What are mutations? So mutations are heritable what in genetic information? What does the mutation mean has happened? It's a change. It's a heritable. Heritable means you can inherit it. So it's something you can inherit, and it's a change in genetic information. Mutations that involve changes in one or a few nucleotides are known as what? Do we know? This is a big one. So, well, this isn't the big one. Sorry, I thought it was. This is point mutations. If a gene in one cell is altered, the alteration can be passed on to every cell that develops from the original one. Now, this can happen during the production of sex cells. It can also happen later in life to any particular cell that is then going to multiply and produce copies of itself. So, let's talk about point mutations. In a what is it when one base is changed? So this is a substitution when one base is changed to a different base. And then you have insertions and deletions, which are point mutations in which one base is inserted or removed from a DNA sequence, and it changes the reading frame. So let's look. Here's an example. Let's say this is an analogy, okay? So let's look at the word beast. B-E-A-S-T. You see it? You see beast? Let's say that's our normal code. A substitution might be feast because what has changed? The B was changed to an F, right? So now beast has turned into feast. That's an example of a substitution. An insertion might be taking beast and adding a letter R and you get breast, and there's a chicken breast, right? So now we went from beast to breast. How about deletion? Well, now we've taken out the A, so now we have best instead of beast. And then lastly, you can have an inversion where letters get flipped. So now instead of beast, we have beats. See it? Cool. So these are different ways you can have point mutations affect your DNA. There are 4 types of chromosomal mutations. So this involves the whole chromosome, not just the coding on the DNA, the whole chromosome. And these are deletion, duplication, inversion, and translocation. So deletion involves a loss or a part of a chromosome. Look right here. This chromosome lost a whole piece. See it? It's missing this chromatid. It's not there. So you can also have duplication, an extra copy or a part of a chromosome. Can you see how this little series of alleles right here on this one has been duplicated? You have twice as many over here, right? This guy, this section was duplicated. You can have an inversion, and this is when it reverses. Do you see how these are flipped? It's the big dark band at the top and then the green. And then here you've got the dark band at the bottom and the green. And then you can also have a translocation, and this is when one part of a chromosome breaks off and attaches to another. So These guys got flip-flopped and came over here and attached to these other guys. So these are all different alterations that can be made to your genetic code. So how do mutations affect genes? The effects of mutations on genes can vary widely. Many have no effect or little effect at all, and some produce beneficial variations. Some negatively disrupt gene function. Cellular machinery that replicates DNA inserts an incorrect base roughly once every 10 million bases. So small changes in genes can gradually accumulate over time. So sometimes it's no big deal. Sometimes it can, over time, get bigger and bigger. So some mutations arrive from what we call mutagens, which are chemical or physical agents in the environment that cause a mutation in a gene, either through affecting the chromosomes or affecting the coding of the gene itself. And we call these epigenetic. That's a good word. So some are chemicals, pesticides, cigarette smoke. You know how you can get cancer from cigarette smoke? Because there are certain chemicals in that cigarette smoke that can affect the genetic code in that particular cell, and then that cell, if it tends to be the code that says, hey, don't stop dividing, don't stop growing, then what happens when cells don't stop dividing and don't die? They just keep dividing. What do you get? Cancer. That's how you can get cancer from any of these things, right? So environmental pollutants, nitrates. Then you can have physical mutagens like X-rays, UV radiation. Do you know people that used to calibrate X-ray machines back when they were invented would take an X-ray of their hand every morning before we realized that they were dangerous? And those people ended up sometimes having their hands almost fall off, like pieces of their fingers would fall off. Gross. What does UV radiation cause if you get too much UV radiation? Cancer again. Skin cancer from the sun. And some cancers, for example, are the product of mutations that cause uncontrolled growth of cells, and sometimes they can come from infectious agents. Certain bacteria or certain viruses can cause cancer too. So let's see. Do I have time for this one? I'm going to come back to this if I have time for it. Otherwise, y'all should watch it independently. So gene, right? Let me pop these in the oven real quick. Come right back, 2 seconds. I'm gonna make sure these are warm. Too warm, Lila? Easy now. All right. Warm donuts are much better, right? Okay. So what controls the development of cells and tissues in multicellular organisms? They're called master control genes. And I just want to show you these really quickly. So for example, there are genes that control how an embryo develops. So we all start out as a single cell, and by 4 days, we're a big old clump of cells like that called a morula. And then the cells start to sort of differentiate. You have some that make like the outside of a ball, and then they all kind of gather in one end, and then they further differentiate into different layers, and these different layers in embryonic development become different types of body tissues, and it's really cool actually to study. So and you guys know about metamorphosis, right? A caterpillar becoming a butterfly. Those are specific types of genes that turn those processes on and off. There are some that are temperature sensitive. Did you know these Himalayan rabbits, if they are reared as babies in less than 20 degrees Celsius, they end up with black points. If they're above 30 degrees Celsius, they're all white. So that depends on temperature. Same thing happens with turtles. A lot of turtle eggs, depending on what temperature they're incubated at, they're either male or female, and that has to do with genes too. And then you have environmental factors. And for example, hydrangeas, you've seen these flowers, right? They can have a different color depending on how acidic the soil is because it's turning on and off certain genes. We're going to skip that one, but I'll come back to mutations. So you saw the video, right? You guys watched the video? Do you remember what stuck out to you? Did you have anything interesting that stuck out to you? No? All right, let's watch the mutations one, and then we'll be done. One.
Since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random, but there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication, an interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched, insertion, which means an extra base or bases are added in. There's also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have three bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome.
Okay, we'll pause there because that's pretty much all you all need to know for that. And you can watch the rest of it at home, but it's also time for lab. So let me get all these lab people.